Am I the only one that didn't know you could stress cancer cells to oblivion? It sounds almost counterintuitive, right? We're so used to hearing about treatments that aim to slow down or stop cancer cells from growing. But what if the key to defeating cancer lies in pushing these cells to their absolute breaking point? Stick around, because this is groundbreaking. This new approach could revolutionize the way we think about cancer treatment. Imagine a world where instead of trying to halt the relentless march of cancer, we force it to run so fast that it collapses under its own weight. Imagine fighting cancer not by stopping it, but by pushing it to its limits. This concept is as radical as it is fascinating. It's like taking the fight to the enemy, making them exhaust all their resources until they can no longer sustain themselves. Researchers Matthias Henrique Dias and Marcelo Santos da Silva have discovered a game-changing approach. Their work is not just a small step forward, it's a giant leap in the field of oncology. They've turned conventional wisdom on its head and opened up a whole new avenue for cancer treatment. Back then, we discovered that a gene known as FGF2 inhibited tumor cell multiplication instead of stimulating it, Dias explained. This was a surprising revelation. FGF2, or fibroblast growth factor 2, was thought to promote cell growth, but in cancer cells it did the opposite. This unexpected behavior of FGF2 was the spark that ignited their innovative research. This unexpected twist led them to think outside the box. They began to wonder if other mechanisms could be exploited to turn cancer's own biology against itself. What if, instead of trying to suppress cancer cells, they could be driven to a point where they self-destruct? Instead of using conventional drugs to halt tumor growth, they hyperactivate the cancer cells. This means pushing the cells to a state of overdrive, where they are forced to work so hard that they eventually burn out. It's a bit like making a machine run at full speed until it breaks down. Picture stepping hard on the accelerator of a car until the engine overheats. That's exactly what they're doing to the cancer cells. By pushing these cells to their limits, they create a situation where the cells can no longer sustain their rapid growth and start to fail. That's exactly what they're doing to the cancer cells. This method is not just about pushing the cells to their limits, but also about making them more vulnerable to subsequent treatments. Once the cells are in this hyperactive state, they become more susceptible to other therapeutic interventions. Once the cells are stressed, they introduce a second drug to attack these vulnerable cells. This two-pronged approach ensures that the cancer cells are not only pushed to their limits, but are also targeted when they are at their weakest. It's a strategic assault on cancer from multiple angles. This dual-drug strategy has shown promise in colorectal cancer models. The results from these initial studies are incredibly encouraging. By hyperactivating the cancer cells and then hitting them with a second drug, they've managed to achieve significant reductions in tumor size. When tumor cells are hyperactivated, they replicate DNA even faster than usual, but their DNA is damaged, Silva noted. This rapid replication under stress leads to errors in the DNA, making the cells even more unstable and prone to failure. This is called replicative stress. It's a state where the cells are under so much pressure to divide and grow that they start making mistakes. These mistakes accumulate, leading to the eventual breakdown of the cancer cells. Their trials in mice showed significant success. The hyperactivation strategy, combined with the follow-up drug treatment, resulted in a dramatic reduction in tumor growth. These findings are a beacon of hope for future cancer therapies. And the best part? This approach could work for other aggressive cancers like pancreatic and bile duct cancers too. These types of cancers are notoriously difficult to treat, but this new method offers a glimmer of hope. Dias and Silva's research is gaining traction. The scientific community is taking notice, and their work is being recognized for its potential to change the landscape of cancer treatment. Large corporations are investing in signaling activators. We hope one of them is ours, Dias said. The interest from big pharma and biotech companies is a testament to the promise of this new approach. With the right backing, this research could move from the lab to clinical trials and eventually to widespread use. This method could even extend to treating parasitic diseases. The principles of hyperactivation and subsequent targeting could be applied to other areas of medicine, opening up new possibilities for treating a range of conditions. Stay tuned, because the future of cancer therapy might just be about stressing tumors to oblivion. This innovative approach is still in its early stages, but the potential is enormous. As research progresses, we could see a new era in cancer treatment, one where we turn the tables on cancer and use its own biology against it. The journey is just beginning, and the possibilities are endless.